everyone and welcome to Miss Estruch Biology. I'm Miss Estruch, a biology teacher and tutor here to help you get to grips with studying biology, improving your exam technique and help you get the grades that you deserve. So if you are new here, click subscribe so you don't miss out on any of the content. So today's video is specifically for exams coming up in 2022. You've had the topic lists, so what this video is for paper 2 A-level biology AQA, going through all the topics that are in the topic list in rank order. Just to let you know as well, I've released 3 new revision products this weekend. We have number 1, the complete A-level active recall workbook, designed for you to consolidate your learning and to revise before you start doing the past paper questions. Number 2, you have your complete guide to creating a revision timetable. I give you a template that you can edit, as well as guiding you through how to use it and how to plan your timetable to maximise your time, and tips to try and make it easier to follow. And finally, the big one, the complete revision bundle. This includes all of my top products that are designed to help you achieve those top grades. It includes the full A-level set of notes, the full active recall workbook, and the revision timetable guide. So if you are serious about improving your grades and you want to get started now, that's the one I'd recommend for you. But for now, let's get into it. Is homeostasis, and this involves physiological control systems to maintain your internal environment within set restricted limits. And the examples that you need to know are maintaining the core body temperature and maintaining the blood pH as well in relation to enzyme activity. Now for those two, you don't really need to know much of the details. It's just knowing that those two conditions are controlled, and you'd need to know why in terms of the effect it has on enzyme activity. Homeostasis involves negative feedback, and this is when any deviation from the normal values, so if it goes too high or too low, there are mechanisms put in place to bring the conditions back to their original level. And it involves the nervous system and often hormones as well. 2 key examples you need to know are blood glucose concentration and water potential. So we'll start with the blood glucose. Now your blood glucose concentrations will increase after you have eaten food containing carbohydrates or drinks containing carbohydrates, and they'll decrease if you haven't eaten food containing carbohydrate for a while or if you have been exercising. The pancreas is the organ that detects these changes in blood glucose levels, and within the pancreas there are cells called the islets of Langerhans, which are responsible for releasing the insulin and the glucagon, which are the 2 hormones that bring blood glucose levels back to normal. Insulin is going to be released when blood glucose levels are too high, and that will bring blood glucose levels down to normal. Glucagon is released when the blood glucose levels are too low. So think of glucose gone, glucagon. And this will cause an increase in the blood glucose levels. Adrenaline, which is released by the adrenal glands, which are found just above the kidneys, this hormone is released when your body anticipates danger, and this results in more glucose being released from the hydrolysis of glycogen in the liver. Now just to look at this as an overview, so your normal blood glucose levels, after you've eaten, they will increase. And this will be detected by the beta cells in the islets of Langerhans in the pancreas. The beta cells will release insulin. That will cause the liver cells to become more permeable to glucose, and there'll be enzymes activated to convert the glucose that's been absorbed into the liver cells into glycogen. That glucose then has now been removed from the blood and stored as insoluble glycogen, and therefore the blood glucose levels have been restored to normal. Now, if you haven't eaten food containing carbohydrates or you've done a lot of exercise, your blood glucose concentration will decrease. This is detected by the alpha cells in the islets of Langerhans in the pancreas, and it will cause the alpha cells to release glucagon, and also the adrenal glands will be releasing adrenaline. The second messenger model occurs to activate enzymes to hydrolyze the glycogen into glucose. That glucose is released back into the blood, and it brings those blood glucose levels back to the normal range. So we're going to have a look at some of those elements in more detail. Firstly, the action of insulin. Now, there's three things that insulin does to reduce the blood glucose levels. First of all, it can attach to receptors on the surface of target cells, so muscle cells and the liver. This changes the tertiary structure of channel proteins within that membrane on the target cell, which means that more glucose is absorbed into the cell by facilitated diffusion. And in that way, the glucose has been removed from the blood and it's now in the cells. There'll also be more of these protein carriers incorporated into the cell membrane, so more glucose can be absorbed. And it activates enzymes so that glucose that has been absorbed into the cells is converted to glycogen. And that is what we mean by glycogenesis. So this is just demonstrating what we said. The insulin binds to the receptors on the target cell, so the liver, for example, and that is going to cause chemicals to be released. And within the cell, there are vesicles that contain the glucose channel proteins, and they move up and fuse with the membrane. So you then have more of those protein channels embedded within the membrane, so more glucose can be absorbed. The action of glucagon, again, there's three different actions. It will attach to the receptors on the target cells, and that would be the liver cells or the muscle cells. And when it binds, it causes the protein to be activated into adenylate cyclase. And this causes ATP to be converted into cyclic or cyclic AMP. That activates another enzyme, protein kinase, and that can then hydrolyze glycogen into glucose. So that is your second messenger model, how you have this two stages of activation of enzymes until we eventually get the hydrolysis of glycogen. It will also activate other enzymes to convert glycerol from lipids and amino acids from proteins into glucose. So this is the second messenger model. The glucagon binds to the glucagon receptors on the target cell, so for example the liver. That, once it is bound, causes this protein to change shape. And that change in shape to the enzyme adenyl cyclase or adenylate cyclase makes it activated. And the activated form can catalyze the reaction of converting ATP into cyclic AMP. And cyclic or cyclic AMP is the second messenger. So the cyclic AMP, which is this second messenger, is triggering the final stage to happen. It can convert the inactive protein kinase into an active form, and that is the enzyme which can then hydrolyze glycogen into glucose. Now, this second messenger model happens for glucagon binding, but also adrenaline. So it's exactly the same process when adrenaline binds. And that's the last thing we've just got here, the summary of the second messenger model, if it was adrenaline binding, but it's exactly the same for adrenaline as it was for glucagon. So pause that if you want, and then you can write down all of those key terms. The liver is involved in all three of these processes. Glycogenesis is when glucose is converted to glycogen, and this occurs in the liver and is catalyzed by the enzymes there. Glycogenolysis or glycogenolysis, pronouncing it like that so it's easier to remember, the lysis of glycogen is the splitting or hydrolysis. And again, this happens in the liver, and this is due to the enzymes involved in the second messenger model. Gluconeogenesis is creating glucose from other molecules, so the amino acids and glycerol, and again this happens in the liver due to the enzymes found there. Now, diabetes is when the body cannot produce enough insulin or it's not responding to insulin anymore. Type 1 diabetes is when people are unable to produce insulin or not producing enough. It normally starts in childhood and as a result of an autoimmune disease where the beta cells are attacked. So the treatment would be injections of insulin to counteract this. Type 2 diabetes, though, is when the receptors on the target cells lose their responsiveness to insulin. It usually develops in adults as a result of obesity and poor diet, and it's normally therefore controlled by regulating the intake of carbohydrates in your diet, increasing exercise, And sometimes it still involves insulin injections also. Osmoregulation is how the water potential of the blood is controlled. Blood with too low a water potential would be described as hypertonic. Blood with too high a water potential would be hypotonic. And if you had hypertonic blood, that means that too much water will leave the surrounding cells and move into the blood by osmosis, and that would cause your cells to shrivel or crenate. If you had hypertonic blood, that means that the excess water from the blood would move into the cells by osmosis, and it could actually cause the cells to burst. Now, neither of these are good, which is why we have to regulate the water potential. What could cause this is if you had hypertonic blood, that could be from excess sweating, not drinking enough, having a diet that's containing lots of ions, so salty diets. If you had hypotonic blood, then maybe you've drunk too much water or you don't have enough salt in your diet. So the corrective mechanisms for both. If you have hypertonic blood, more water has to be reabsorbed by osmosis into the blood from the tubules in the nephron, which is in the kidney. Therefore, you'll have urine which is lower in volume and more concentrated. If you have too much water in your blood, so hypertonic, that means less water will be reabsorbed by osmosis into the blood from the tubules in the nephrons in the kidneys. And as a result, you'll be producing larger volumes of urine, which are more dilute. Now, all of this happens in the nephron, and you need to know the structure of the nephron, which you find in the kidneys. The nephrons are these long tubules. They're surrounded by capillaries, and you have around 1 million of these in both of your kidneys. It's where the blood is filtered to remove the waste, but also selective reabsorption happens to reabsorb any useful substances back into the blood. So the way this happens is, step one, as the blood is going to flow into the glomerulus, the capillaries there, because there's a really, really high hydrostatic pressure, water and small molecules are forced out of the capillaries and into the renal capsule, which is this yellow part here, and the glomerulus is all of the red bit there where you have the capillaries. You've then created this filtrate, and that filtrate will then start to pass through to the proximal convoluted tubule. And this is where selective reabsorption occurs. So glucose is reabsorbed in the proximal convoluted tubule. The loop of Henle is where we maintain a sodium ion gradient, and that is to make sure that water can then be reabsorbed back into the blood. So at this point, the filtrate is going to be losing lots of the ions in the ascending limb, and that will then lower the water potential of the surrounding area around the loop of Henle, and then water will be able to move out by osmosis as it goes down the descending limb. So as the filtrate reaches the distal convoluted tubule, it's lost a lot of water, but also quite a lot of ions. And because it's lost a lot of ions, it is actually quite dilute by this stage. So more water will move out by osmosis, and at the collecting duct, even more water can move out to go back into the blood. The collecting duct carries the remaining liquid away to form the urine. Now that is a very basic overview. To have the full details, you will need to watch my full video where I actually go through every one of those steps in a lot more detail. This is just the basic summary video. That would not be enough detail though, so do check that one out up here. So how then this is regulated is using the hypothalamus and the posterior pituitary gland in the brain. The hypothalamus is where the changes in water potential are detected by receptors called osmoreceptors. If the water potential of the blood is too low, water leaves the osmoreceptor cells by osmosis, and that causes them to shrivel. And this stimulates the hypothalamus to produce more of a hormone called ADH, or antidiuretic hormone. If the water potential of the blood is too high, water enters the osmoreceptors by osmosis. This stimulates the hypothalamus to produce less antidiuretic hormone. So the hypothalamus is where the ADH is produced, but it then moves to the posterior pituitary, and from here it is released into the capillaries and into the blood. ADH travels through the blood and to its target organ, which is the kidney. Now, what that hormone does is it causes an increase in the permeability of the walls of the collecting duct and the distal convoluted tubule, so they're more permeable to water. And as a result, more water will move out of the nephron by osmosis and it's reabsorbed into the blood. So you'll have more water returning to the blood and more concentrated urine. So we just have a look at this summary then. If you are going to move away from the normal water potential and therefore you have water potential of the blood increasing, meaning you have too much water, that's going to be detected by the osmoreceptors in the hypothalamus. The hypothalamus will release less ADH. That will cause the distal convoluted tubule and the collecting duct walls to become less permeable to water. So less water is reabsorbed into the blood and more is lost in the urine. So you'll have larger volumes of more dilute urine, and that will bring your water potential of the blood back within the normal range. If you have very concentrated blood or the water potential is more negative, that again will be detected by the osmoreceptors in the hypothalamus. The hypothalamus will release more ADH, which is released into the blood by the posterior pituitary gland. The DCT and the collecting duct walls become more permeable to water, so more water is reabsorbed into the blood and less is lost in the urine. And that will bring your water potential back within the normal range. Next then we have a look at respiration, starting with aerobic respiration. And there are 4 key stages: glycolysis, the link reaction, Krebs cycle and oxidative phosphorylation. And for each stage, you need to know exactly where in the cell it occurs. And that's what we have in brackets here. So we'll start with glycolysis. This does produce some ATP, but not a lot. And it also happens in anaerobic respiration because oxygen is not required for glycolysis. It happens in the cytoplasm, not actually inside of the mitochondria. And there are 3 key stages that AQA say you need to know. First thing that happens is that glucose is phosphorylated to glucose phosphate, and that uses 2 molecules of ATP. That glucose phosphate very quickly is converted into triose phosphate, and then triose phosphate is oxidated to form pyruvate, and that produces 4 ATP and reduced NAD, which is the coenzyme used in respiration. So let's just have a look at what that would be on a diagram. Again, the yellow circles are representing carbon. So glucose is a 6-carbon compound. We said the first thing that happens is glucose is phosphorylated, and what that means is phosphate groups are added to it. Now we're going to add 2 phosphate groups, so we need 2 ATP molecules, and that creates glucose phosphate. That is an unstable compound, so it splits into 2 3-carbon compounds called triose phosphate. And then the triose phosphate, or TP, both of those molecules are oxidized. Both TP molecules will result in the gain of 2 ATP when pyruvate is made, and also as each is oxidized, there'll be a reduced coenzyme created. So NAD picks up a hydrogen to form NADH, or reduced NAD. So at the end we have 2 molecules of pyruvate, a net gain of 2 ATP, and what we mean by that is, although we've made 4 ATP molecules in this stage, we had to use 2 at the beginning. So overall, we've created 2. We've also made 2 reduced NAD. So that pyruvate that has been created and the reduced NAD, they are needed in the next stages, which is within the mitochondrial matrix. So they have to be actively transported into the mitochondrial matrix, ready for the link reaction. So the pyruvate is going to be oxidized further to make acetate. We're going to have an NAD picking up a hydrogen to create yet another reduced NAD molecule. So that's the first step we can see here. Pyruvate, our 3-carbon compound, it's going to be oxidized further, and we can tell it's been oxidized because it's lost the hydrogen, which has been picked up by the coenzyme NAD. So that has created acetate. Acetate is only a 2-carbon compound, and that is because carbon dioxide is produced at this stage of the link reaction. Now that's not the end, because the acetate then combines with another coenzyme called coenzyme A, and that creates acetyl-CoA. And acetyl-CoA is what can then enter the Krebs cycle. So overall, for every glucose molecule, there are 2 pyruvates created. So for every glucose molecule, the link reaction happens twice. So for one glucose molecule, we'd create 2 acetyl-CoA, 2 carbon dioxides, and 2 reduced NAD. So that acetyl-CoA can then enter the Krebs cycle, and it combines with a 4-carbon molecule. And that will then create a 6-carbon molecule. Now the coenzyme, its only job is to deliver the acetate to that 4-carbon compound. So once that happens, the coenzyme is released and it can be reused in the link reaction. We then have a series of redox reactions, and the Krebs cycle is going to be generating even more reduced coenzymes, a small amount of ATP, and some carbon dioxide. And that's what we can see in this stage here. The 6-carbon to the 4-carbon molecule, which is then regenerated. To go from 6 to 4, we must have lost 2 carbons, and that's in the form of 2 carbon dioxides. We only have 1 molecule of ATP being produced, but we have quite a few reduced coenzymes. We create 3 reduced NAD and 1 reduced FAD, which is our final coenzyme involved in respiration. So overall, the products per cycle are 3 reduced NAD, 1 reduced FAD, 1 ATP, and 2 carbon dioxides. But for every glucose, the cycle happens twice because we'd have 2 acetyl-CoA produced, so 2 rounds of the cycle. So we actually double all of those values. So look carefully at an exam question, whether they're asking for the products per cycle or per glucose molecule. Oxidative phosphorylation is the final step, and this is very similar to what we looked at in chemiosmosis in the light-dependent reaction of photosynthesis. It's when most of the ATP is produced. So the first thing that happens is in the mitochondrial matrix, all of those reduced coenzymes that were produced are going to release their hydrogen, and that splits into protons and electrons. Those electrons are going to be transported along the electron transfer chain. That releases energy, and that energy is used to actively transport the protons from the mitochondrial matrix into the intermembrane space. That creates this electrochemical gradient, and therefore the protons can move by facilitated diffusion down their concentration gradient through ATP synthase. And the ATP synthase will then phosphorylate ADP to create ATP. So that is how we create lots of ATP in this stage, and you actually create 34 ATP molecules. Now at the end of the electron transfer chain, those electrons are picked up by oxygen. And oxygen also picks up the protons once they've passed through. And that is how we create water. So we say that oxygen is the final electron acceptor, and it will form water when it also picks up those protons. Anaerobic respiration is without oxygen, and it only occurs in the cytoplasm. In plants and microbes, anaerobic respiration produces ethanol and carbon dioxide. In animals, it produces lactate or lactic acid. And this stage is all to do with reoxidizing NAD so that glycolysis can continue to occur. So what we have happening is glycolysis occurs as usual because no oxygen is needed for that stage. But instead of the pyruvate being transported into the mitochondrial matrix for the link reaction, it remains in the cytoplasm, and the pyruvate is reduced to form lactate. We know it's reduced because the reduced coenzymes that were just produced in glycolysis are immediately reoxidized to NAD, and that is because the pyruvate gained the hydrogen. Now that is important because that NAD can then be reused in glycolysis so that glycolysis can continue to occur. And that ensures that at least some ATP is continually made. So the downside is lactic acid is acidic, so it can denature enzymes and other proteins, but that is the price to pay so that at least some respiration can still occur. But you can't respire anaerobically for long because that buildup of lactic acid will eventually denature the enzymes involved in glycolysis. Same idea for microbes, except we have ethanol and carbon dioxide produced. Again, ethanol is going to be harmful, but it's to ensure that glycolysis can still continue, so at least some ATP is being produced for metabolic reactions to continue. Now, if we have a look at a myelinated motor neuron structure, the key parts are, first of all, the cell body. And this cell body contains all the organelles you'd have in a typical plant cell. So, for example, we can see the nucleus. It also has proteins and neurotransmitter chemicals which are made in that part of the cell. The dendrites we can see here branching carry action potentials to surrounding cells. The axon is the conductive long fibre that is making up this whole part of the cell, and that carries nerve impulses all the way along this particular motor neuron. Schwann cells are what we call these layers wrapping around the axon, and they form the myelin sheath, which is a lipid, and therefore it insulates. It does not allow the charged ions to pass through to the axon. There are gaps though, and these gaps are called the nodes of Ranvier, and that is where they are not insulated. A resting potential is what we would have happening inside of the axon when there is no stimulus. So when a neuron is not conducting an impulse because there's no stimulus, there's no change in the environment, there is still a difference between the electrical charge inside the neuron, so inside the axon, and outside. And we call that difference the resting potential. There are more positive ions, the sodium ions and potassium ions, outside compared to inside of the axon. And therefore the inside of the neuron is comparatively more negative. And that is why the resting potential is minus 70 millivolts. Now you need to know how that resting potential is established in the first place after a response to a stimulus, and how it's also maintained if there is no stimulus. Now the key way it's maintained is by a sodium-potassium active transport pump. So this is an example of co-transport, but it is also active transport because ATP is involved. And at this pump you have 2 potassium ions being pumped into the axon, and 3 sodium ions are being actively transported out of the axon. This creates an electrochemical gradient, and therefore the potassium ions, because there's a higher concentration of them inside the cell, they will start to diffuse out of the cell. The sodium ions, there's a higher concentration outside, so they will start to diffuse into the cell. However, the membrane is more permeable to potassium ions because there are more potassium ion channels and because some of those ion channels remain permanently open. So that means more potassium ions can diffuse out than sodium ions diffusing in. And as a result, there are more positive ions outside of the cell compared to inside of the cell, and that's how we get the minus 70 millivolts. Now, if there is a stimulus, and if it's a large enough stimulus, it can generate an action potential. An action potential is when the neuron's voltage increases beyond a set point from the resting potential. And this is what generates the nerve's impulse to then travel along the neuron. So an increase in voltage, also known as depolarization, is due to the neuron potential becoming more permeable to sodium ions and therefore becoming more positive. Once an action potential is generated, it moves along the axon like a Mexican wave. So let's have a look at then how this action potential is generated. A stimulus provides the energy that can cause the sodium ion voltage-gated channels in the axon membrane to open, and this will cause the sodium ions to start diffusing in. And that's what we can see here. One of those channels has now opened, and therefore sodium ions will start to diffuse into the axon. The potassium ion channel here is permanently open, but because we now have some sodium ions moving in, we start to see this increase in the voltage. Now, if the stimulus is large enough to cause enough of these sodium ion channels to open, then you'll have enough sodium ions diffusing in to cause an increase beyond the threshold potential. And if you do have an increase beyond that minus 55 millivolts, then you will always have the rest of this curve happening. You'll always have an action potential generated. Now, there are even more voltage-gated sodium ion channels. So as that voltage will start to increase, that will cause more sodium ion channels to open. Then we have this positive 35 to 40 millivolts. What then happens though is we see this decrease, and that's because when it does reach this particular voltage, it then causes the voltage-gated sodium ion channels to close. So no more positive ions are entering, but we still have those potassium ions moving out. So we start to get this decrease, which we call the repolarization. That will then cause more potassium ion channels to open, so even more potassium is diffusing out. And eventually, so many positive ions have diffused out, we get to that resting state, but actually it overshoots and goes to minus 80 millivolts, which we can see here. We're now back to that original state of potassium ions would be able to move out, but all of the other gates are closed. So that is the generation of an action potential. And this happens along the axon multiple times. This graph is not a change over distance. So this isn't happening just once along the axon. It happens right at the start of the axon, and then at every node of Ramvir, if it is a myelinated neuron, you'll then have another action potential being generated until you get to the end of that neuron and then a synapse. So it's a bit like the idea of a Mexican wave moving along. If it was unmyelinated, then same idea, but it has to happen at every single position on the axon because you wouldn't have the myelin sheaths. So that would mean it would take longer for all of those action potentials to finally reach the end. Now you need to know about this all or nothing principle, and it links to the threshold. So if the axon isn't depolarized to at least minus 55 millivolts, which is the threshold to cause the rest of the action potential to be generated, then the impulse will not be produced. So what we mean by the nothing part of the principle is if the stimulus isn't large enough to cause a voltage increase to at least minus 55 millivolts, then nothing more will happen. And that's what this bit is showing you here, some failed initiations of an action potential. The all part of the principle is the fact that if you do exceed that threshold, then you will always have an action potential and it will always peak at exactly the same point. So even if the stimulus is bigger, so brighter light compared to dimmer lights, you will still have the same peak. But what changes is the frequency of those action potentials being generated. So that is the all or nothing principle, and the reason it's important is to make sure that animals are only responding to stimuli which are large enough rather than responding to every single slight change in the environment. Because if we did that, it would actually overwhelm your senses and it wouldn't necessarily be protecting you from stimuli which might be causing you danger. Now, the refractory period we mentioned is when it goes into this minus 80 millivolts. But the importance of this is, once we have this stage, we have a period when another action potential can't be generated straight away. And that's because these sodium ion channels, we describe as they're recovering. But essentially what we mean is, because we've now gone even more negative, you would have to have even more sodium ions to be able to reach that threshold. The importance of this is, first of all, it ensures that discrete impulses are produced. So an action potential cannot be generated immediately after another, and that makes sure each one is separate. So that helps with you being able to identify the source of the particular stimulus. It also makes sure that the action potentials will only be travelling in one direction along the axon. So it stops the action potential from spreading out in both directions, which would actually result in the impulse not travelling along. It limits the number of impulses as well, and this is important to prevent you overreacting to a particular stimulus or over-responding. Now, the factors that will affect the speed of conduction are myelination and saltatory conduction, the axon diameter, and temperature. So the first one was myelination, and that results in saltatory conduction. And what that term means is the action potentials will jump from node to node, meaning the node of Ranvier. And that is because this myelin sheath created by the Schwann cells is insulating, so the ions cannot diffuse in and out during these sections, so it jumps to the parts where there is no insulation. Now, as a result, that means that the impulse will travel much faster along the axon because you don't have to generate as many action potentials. You're only generating them at those nodes. Next then, the axon diameter. The wider the diameter, that is going to increase the speed. And the reason for that is basically like the idea of physics and wires and electricity. So the wider the diameter, that means we're going to have less leakage of the ions, and therefore the action potential can move faster. Temperature, there's two reasons why this has an effect. Number one, because if you have a higher temperature, the ions will have more kinetic energy. That means they'll be able to diffuse faster. The second reason is the enzymes involved in respiration to provide ATP for the sodium-potassium pump will be able to work faster as well if there is a higher temperature. Now, if it does exceed the optimum of around 37, then that could actually decrease the speed. So synapses are the gaps that we have between the neurons. So once you have your action potential reaching the end of a neuron, it has to get to the next one. And in order to do that, we have to have neurotransmitters, which are chemicals, diffusing across the gap, which will then generate the action potential in the next neuron. Now, the way that this happens is, first of all, your action potential, as I said, it will arrive at the end of the neuron, and we describe that as the synaptic knob. Depolarization of the synaptic knob leads to calcium ion channels opening, and therefore calcium ions will diffuse into the synaptic knob. Those calcium ions will cause the vesicles that contain the neurotransmitter to move towards the membrane, fuse, and release the neurotransmitter into the synaptic cleft. Because we have a high concentration of the neurotransmitter where they've just been released, they'll then diffuse across to the postsynaptic membrane. And on the postsynaptic membrane, there are receptors, complementary in shape, for those neurotransmitters to bind to. When those neurotransmitters bind to the receptors, it causes the sodium ion channels on the postsynaptic membrane to open. Therefore, sodium ions will diffuse in, and if enough sodium ions diffuse in to exceed that minus 55 millivolt threshold, then the action potential is generated. So that will then cause that action potential to be generated in the postsynaptic membrane, and then it can continue along the axon of the next neuron. Now, this is unidirectional, and you could be asked, how is it ensured that it's unidirectional? And there's a couple of ideas. Number one, the vesicles containing those neurotransmitters are only in the presynaptic neuron, so they can only be released from this side. Because of that concentration gradient, diffusion happens from the pre to the postsynaptic neuron, and you only find the receptors complementary in shape to those neurotransmitters on the membrane of the postsynaptic neuron. Now, one example of a synapse that you need to know is the cholinergic synapse. So it's exactly the process that we just went through, but you need to know the name of the neurotransmitter. So the neurotransmitter is acetylcholine. It will bind to its receptor, and then it will go through the whole stages that we said. But those neurotransmitters don't remain permanently bound. If they did, that would mean the sodium ion channels would be continually open, and you'd continually be triggering this action potential and a response, even though the stimulus wasn't present anymore. So to prevent that happening, there is an enzyme that will break down the acetylcholine into choline and acetate, which can then be reabsorbed into the presynaptic neuron and be reused. And that neuron is acetylcholine esterase. Now, I said that in order for the action potential to be generated in the postsynaptic neuron, you needed to have enough sodium ion channels opening, and that is caused by the neurotransmitters binding to the receptor. And there's two different mechanisms of summation to ensure that happens. Summation is referring to the fact that you have this rapid buildup of neurotransmitter to generate the action potential, and you can either have spatial or temporal. Spatial is when you have multiple neurons connecting to the same postsynaptic neuron. And collectively, these three neurons in this example should release enough neurotransmitter to cause enough of those sodium ion channels to open on the postsynaptic neuron. The alternative is temporal, and this is where you only have one presynaptic neuron connected to one postsynaptic neuron, but instead the way that this will cause enough neurotransmitter to be released is you have repeated releasing of that neurotransmitter over a short period of time, until eventually you have enough neurotransmitter bound to enough of the receptors on the postsynaptic neuron's membrane to open enough of those sodium ion channels. You also have inhibitory synapses, and in this case you would have chloride ions would be moving into the postsynaptic membrane. So you'd have different neurotransmitters binding to their complementary receptor, but this time it would cause chloride ion channels to open. And because you'd then have negative ions moving in, this would cause the membrane potential to decrease to at least minus 80 millivolts, and therefore the postsynaptic membrane would go into this hyperpolarization, making it very unlikely that an action potential could be generated. Neuromuscular junctions are what you have right at the end of your response arc. So you have your motor neuron, you then have a gap, and then you have your muscle. And your muscle is one of the effectors that would generate the response. And what you need to know is similarities and differences between the synapse that you find here at the neuromuscular junction compared to your typical synapse such as the cholinergic. So what they have in common is both of them will work in one direction only, so unidirectional. And that is because the receptors for the neurotransmitters are only found on the postsynaptic membrane, or in this case, the muscular membrane. A neuromuscular junction will only be excitatory, meaning it will only generate that response. Whereas a cholinergic, we looked at, could be excitatory, generating an action potential, or it could be inhibitory, which prevents the action potential from being generated. The neuromuscular junction will have a neuron connecting to a muscle, and it's always the motor neuron. The cholinergic is just a synapse between any two neurons. The neuromuscular junction is always at the end point for the responses, and the cholinergic synapse is always going to be generating a new action potential. For the neuromuscular junction, the acetylcholine will bind to receptors on muscle fibre, whereas the acetylcholine binds to receptors on the postsynaptic membrane in the cholinergic synapse. The next bit in this topic is looking at how energy is transferred in an ecosystem. So it's looking at food webs. And all food webs begin with producers, which are green plants, because they can photosynthesize to use the light energy to make their own organic compounds. So they don't need to eat, they produce their own food. Between each trophic level, which is the term of what we call each stage in a food web, the majority of energy is lost, and that is due to respiration and excretion. The remaining energy is used to form the biomass. And the biomass is the mass of carbon within the organism, or you can describe that as the dry mass. Now that is a key term, so definitely learn that definition. And we give biomass per given area, so it can be a fair comparison between organisms. MPP and GPP are two key terms that you need to know. So how productive an ecosystem is depends on the abiotic and biotic factors that are within that ecosystem. If there's plenty of water, light, it's warm, there's lots of green plants, then you'll have a really high rate of photosynthesis, and that results in more carbohydrates being produced by the plant. And we can quantify this using GPP and MPP. So GPP is gross primary production, and it is the chemical energy stored in the plant biomass in a given area, or it could be volume, and it's the total energy resulting from photosynthesis. NPP is the net primary production, and that is the GPP minus the energy that will be lost by respiration. So basically MPP is the energy you have left to go towards making the biomass. Now another formula that you could be asked to use is this one here, looking at the net production in consumers. So to work out the net production in consumers, so net production of biomass this is, you would need to use the formula N equals I minus F plus R. I is the chemical energy stored in ingested food, so that is the food that's eaten. F is the chemical energy lost to the environment in feces and urine. And R is respiratory losses. So whatever you have left, the N, that can go towards making the carbon compounds or the biomass. The rates of productivity are recorded using the units kilojoules per hectare per year. So there's 3 parts to those units, and it can be asked as a 2-mark question to give the units, or to say why you have 3 parts to the units. So the kilojoules is the unit of energy, but these units also include per unit area and per year. So it's recorded per unit area to standardise the results to enable environments to be compared. So it's taken into account that different environments will vary in size. The units are also per year to take into account the impacts of the seasons. So when you have rain, when it's light or when it's less light, the temperature changes. That's going to affect the productivity because it affects photosynthesis. So we do it per year, so you have an annual average, so it's a fair comparison. And cycles. And we're going to start with the nitrogen cycle. The nitrogen cycle is really important because nitrogen is within proteins, ATP and nucleic acids. So that's the DNA and RNA. So it's essential that animals, plants, microbes have nitrogen within them to create these molecules. And we need this cycle because you can't actually gain nitrogen gas through gas exchange like you can with oxygen or carbon dioxide for plants for photosynthesis. And the reason for that is nitrogen gas has this triple bond, and we don't have the enzymes to be able to break those bonds and then use the nitrogen in compounds. Microorganisms do though, so they have the enzyme to be able to do that, or some of them do, and therefore we need this cycle involving microorganisms to make the nitrogen accessible for nitrogen containing substances, which plants and animals can then use. The nitrogen cycle involves these key processes: ammonification, nitrification, nitrogen fixation and denitrification. But you also need to be aware that saprobiotic nutrition and microbes are essential for all of those stages highlighted in blue. So saprobiotic nutrition is referring to when microbes are feeding on dead plant matter or waste, and it's the microbes that are going to be doing that. So if we have a look at the cycle, we have nitrogen gas in the atmosphere. There'll be nitrogen fixing bacteria that can convert that nitrogen into ammonia. And that will either be done directly by nitrogen fixing bacteria in the root nodules of leguminous plants, which are types of plants like clover and beans, which have these lumps or nodules on their roots that contain bacteria. So for them, the bacteria will directly convert the nitrogen in the atmosphere into ammonium into their roots. Plants that don't have those root nodules, there is free living nitrogen fixing bacteria within the soil, which can also fix nitrogen into ammonium. Then we have nitrification occurring, which is when nitrifying bacteria can oxidize ammonium into nitrites and then oxidize it further into nitrates. And that can then be absorbed by active transport into the plant roots. And then animals can absorb that through digestion and absorption. Now, the decomposers will be breaking down the nitrogen containing compounds in the dead plant and animal material and in the waste produced by animals. But for A level, it's not enough to just say the dead material or waste. You have to name the nitrogen containing compounds. So the waste, you could name urea. And then in the dead plant and animal material, it would be proteins. It could be DNA as well. So you have to name the compound that the decomposers would be breaking down. And this would be our saprobiotic nutrition. So the decomposers will break down the proteins to release the nitrogen, and it converts it back into ammonium, and then the cycle can go round again. If, though, there is a lack of oxygen within the soil, we then have anaerobic denitrifying bacteria taking over, and they can convert the nitrates back into nitrogen gas. So that's how we get the nitrogen going back into the atmosphere. So other than the denitrifying bacteria stage, all of the other stages are oxidation, and they would require oxygen to be present. The other cycle you need to know is the phosphorus cycle. And phosphorus is really important because it's used in the phosphate groups of DNA, RNA, ATP, and in the phospholipids within the phospholipid bilayer. So it is an essential element for life for all organisms. The phosphorus cycle is different to the carbon and nitrogen cycle. Not that you actually learn the carbon cycle at A level. And that is because you do not find phosphorus as a gas in the atmosphere. So instead, the main store or source is as phosphate ions in the mineral form in sedimentary rock. Now you need to know about the mycorrhizae, which are fungal associations which occur between the plant roots and beneficial fungi. Now, although we're talking about this here in the phosphorus cycle, it would also be occurring in the nitrogen cycle as well. The mycorrhizae are going to provide benefits for plant growth. And that's because the mycorrhizae fungal associations, we can see here you get these really long networks of fungi, and they can spread out in the soil to create a larger surface area for the absorption of water and mineral ions going into the roots eventually. The plants provide an advantage to the fungus because the plants through photosynthesis will create carbohydrates, and it can share those carbohydrates with the fungus. So this is a mutualistic relationship because they both provide a mutual benefit to each other for survival. So the phosphorus cycle, if we start in the middle looking at the phosphate ions in the oceans, or it could be within the soil. Plants can absorb those phosphate ions through their root hair cells by active transport. Animals will then be eating the plants, so they can consume and absorb the material to get the phosphate ions. When animals excrete, that will release phosphorus back into the oceans or the soil in the form of phosphate ions. As well as returning it to the soil, we also have decomposition. And phosphate ions from waste and remains are going to be broken down. So animals which have a shell, but also bones, contain phosphate ions. And those can be eroded to release the phosphate ions back into the ocean or the soil. The other thing you can have, which we can see in this image, is guano. And guano is from bird feces, and it's this white layer you can see on the rocks. So it's a type of excrement which is really rich in phosphate ions. Deposition is when we get phosphate ions building up to create the rocks, and that also links to sedimentation over here. So over time, from high pressure as well, the phosphate ions within the oceans, within the soil, will compact through sedimentation to create the sedimentary rocks. And then the last link back is the fact that those sedimentary rocks can be eroded to release the phosphate ions back into the soil or oceans. Now, the extra one added here is the idea of fertilizers. Fertilizers added to soils can also run off from the soil or leach into water sources, so it would return it back into oceans, or it could just be within the soil itself, but that will be adding phosphorus into the cycle. If we just look at fertilizers a little bit further, we said that they can be added to the soil. The reason for that is when crops are harvested, you're removing the plants, and that is therefore removing one of the stores of nitrate or phosphate ions. So it's going to be taken out of the nutrient cycle. And if you continue to do that, eventually you'll have plants and soil which are mineral deficient. So you have to add either natural or artificial fertilizers to replace the minerals and the nutrients that are being removed when you harvest crops. So natural fertilizer would be animal manure. Artificial is soluble chemicals which contain exactly the proportion and quantities of the mineral ions required. Now, the natural fertilizers are cheaper because if you have your own animals on a farm already, it's completely free. It is just the manure from the animals. However, it doesn't contain necessarily the exact proportions of the minerals you need, and you have no control over that. Artificial fertilizers, because they are synthetically created, they are created to have the exact proportion of minerals that plants would require. However, the inorganic substances are very, very soluble, which has the downside of that they can dissolve in the rainwater and leach, which means they're washed away. So those soluble chemicals such as nitrate, phosphorus, dissolve in the water and then they leach, which means they run off from the rainwater into nearby water sources, such as rivers or ponds. If that nitrogen fertilizer leaches into waterways, it causes eutrophication. And eutrophication is when nitrates are going to stimulate the growth of algae in a pond or a river. It's more likely to happen in a pond though, because in a river the movement of the water can prevent the algae from growing or staying in one place. So the excessive growth of algae creates this blanket on the top of the water. That blocks the light. As a result, plants below that blanket aren't going to be receiving light for photosynthesis and they end up dying. Bacteria within the water will then feed and respire on the dead plant matter, and in doing that they're going to be using up the oxygen within the water, and you get more and more bacteria growing and thriving because of all that dead plant material to feed on, so even more oxygen is used up, and eventually there won't be enough dissolved oxygen within the water for animals, And animals such as fish and other aquatic organisms will therefore die because there's no oxygen for respiration. So the first thing with inheritance is there are a lot of key terms that come up quite often as a 1-mark definition just to start off an inheritance question. So make sure you know all of these definitions as they've all come up at some point in the past. So genotype is the genetic constitution of an organism. Phenotype is the expression of the genes, but also the interaction with the environment, so don't forget that bit. Homozygous is when you have a pair of homologous chromosomes carrying the same alleles for a single gene, whereas heterozygous is when you've got the homologous chromosomes carrying 2 different alleles for a single gene. Recessive allele is only expressed if there's no dominant allele present, whereas the dominant will always be expressed. Codominant is when both alleles are equally dominant and expressed in the phenotype. Multiple alleles is when you have more than 2 alleles for a single gene. Sex linkage is a gene whose locus is on the X chromosome. Genes are located on the same chromosome, not the sex chromosomes though, for autosomal linkage. Epistasis is when one gene modifies or masks the expression of a different gene at a locus. And then monohybrid and dihybrid is referring to types of genetic crosses. So monohybrid is when you're looking at the inheritance of just one gene. Dihybrid is when you're looking at the inheritance of 2 genes at a time. And we're going to go through the types of inheritance in more detail and have a look at examples of genetic crosses. Now, a big part of the inheritance topic is being able to determine the outcome of genetic crosses. So to help you with that all in one place, I've got you this genetic coding table. So for the different types of inheritance, this is how you would represent it in a genetic cross. A monohybrid is what you would have done at GCSE. So you pick a letter to represent the gene, capital is the dominant allele, lowercase is the recessive. For codominant, because they're both dominant, you can't go for a capital and a lowercase. So instead, the base letter is representing the gene and the superscript letter is representing the allele. So for example, blood group is IB for blood group B, IA for blood group A, but if you have this here, IBA, that would mean that it's someone that's blood group AB because both dominant alleles will be expressed. Multiple alleles, again, you can't use the upper and lower case because you have more than 2 alleles, and you might have this linked with the idea of codominance as well, as we see in the example of blood groups. The allele that codes for blood group B is dominant, the allele that codes for blood group A is dominant, but there are multiple alleles because there is a third one for blood group O, but that is recessive. For sex linkage, you have to represent this by showing, first of all, the X and Y chromosome, and in sex linkage the gene is only found on the X chromosome, so you'll only be drawing the allele on the X chromosome, not on the Y chromosome, but we still stick to capital for dominant, lowercase for recessive. Autosomal linkage is always linked to dihybrid because it's looking at the inheritance of 2 genes found on the same chromosome. So we can still use the capital and lowercase, but we'd have 2 different letters representing the 2 different genes. Epistasis would be the same idea as well, because they often don't link it to any of the other types of inheritance because epistasis is complicated enough. So now for the examples. So we're starting off with a simple monohybrid example where we're looking at the inheritance of cystic fibrosis, which is caused by a recessive allele. And we're told in this question that we have 2 parents that are heterozygous, so we can see that represented here. And we're being asked what is the probability they will have a girl with cystic fibrosis. Now don't be thrown, this is not a sex-linked gene, but they still want you to work out the probability of, first of all, whether the child will have cystic fibrosis. And we can see here we have 25% probability that a child would have cystic fibrosis. But because they want you to say the probability of a girl having cystic fibrosis, there's 50% probability they'd have a girl. So we'd then do 25% times 50%, or half of it basically. So there's 12.5% probability that they'll have a child who has cystic fibrosis who is also a girl. Now if we have a look at a codominant example, cows can be red, white or roan in colour, and this is roan here where it's blotchy with both colours expressed. And in this example we have 2 roan cows reproducing, and they want to know what's the probability they'll produce red offspring. So 2 roan cows, because this is codominance, both the red and the white are dominant. That's how it's possible to get roan where they're both expressed. So here is how we'd represent it for the 2 parents. And then if we do our Punnett square, we can then see we'd have 1 roan, 1 red, actually a second roan down here, and 1 white. So that would mean we have 25% probability of getting a red cow from those parents. Looking at the inheritance of blood groups is an example of multiple alleles and codominance. So here we have the phenotypes that are possible of the different blood groups. Now you don't need to know this. If they wanted you to work out anything to do with blood groups, they would have to give you some information so you could work it out. It's not on the spec, you have to know the blood groups. It's just a really common example to test your understanding of codominance and multiple alleles. So blood group A, you could either have 2 copies of the dominant A, or 1 copy of the dominant A and 1 recessive O allele. Same idea for blood group B, but with the B alleles. To be blood group AB, this is our codominance where we have 1 copy of both of those dominant alleles, so both are expressed. Blood group O is recessive, so it's only possible if you have 2 copies of IO, which is our recessive allele. So if we have a look at an example, we have parents with blood group AB and O reproduce, and we're told that A and B are codominant, O is recessive, because as I said, you're not expected to know that. It would be application of your knowledge. And they want to know what's the probability they'll have an offspring with blood group A. So the first thing is you have to state the parental genotypes. Then we can work out the gametes, which I've put here on the outside of the Punnett square. Then we have the offspring genotype and the offspring phenotype. So we can see the probability of having blood group A is 50%, because we have 2 individuals down here that have both got a genotype that codes for blood group A. So now our example of sex linkage, it's looking at colour blindness, which again you don't need to know, it's an application of your knowledge, and you're told that colour blindness is caused by a recessive allele found only on the X chromosome. If a non-colourblind male reproduces with a female carrier of the allele, what is the probability their children will be blind? So first of all, we need to work out the parents' genotypes. So the mother is a carrier, so that means they'll have 1 dominant allele and 1 of the recessive alleles that causes colour blindness. And then the male, XY, they do not have colour blindness, so that means they have to have just the dominant allele on their X chromosome. So here we have our gametes. We then have the offspring's genotypes and phenotypes. So the probability that they would have a child that is colourblind is 25%. So epistasis, we said, is when one gene influences or affects, masks the expression of another. So some examples are the coat colour in mice, or in Labradors, fruit colour of vegetables as well, such as squash. And we'll have a look at the Labrador example. So gene 1 controls whether pigment will or will not be expressed, and the dominant allele codes for pigment will be produced, whereas the recessive codes for no pigment, and therefore the dog won't be producing any of the darker pigments and it'll be that yellow colour. Gene 2 controls what colour the pigment will be if it is expressed. So dominant is black, and recessive is brown. So that is how you get the 3 different colours in Labradors. So we have a look at an example. We're going to cross these 2 parents. So the first thing, we've been given the genotypes, but we have to work out the phenotypes. Now both of them have a dominant E, which means pigment can be produced, and they have a dominant B, which means they'll be black. After that, it's working out all the possible gametes that we could have. Then we'd need to do a Punnett square to work out all the possible genotypes of the offspring, and then the phenotypes of the offspring, which I've done for you just here. So we can see we end up with 4 yellow dogs because they have homozygous recessive for that E gene. So even though they might code for black and brown, we won't see that colour because it's been masked by the first gene. Now, dihybrid crosses are when we have 2 genes considered at the same time in a Punnett square. So a common example is Mendel's peas. So we've got round and yellow, wrinkled and green as our 2 different traits, the shape versus the colour. And in all of the genetic crosses that we've gone through, it's these 6 features you have to have for all of the marks. We need the parental phenotype, genotype, the gametes, offspring genotype, and then you match the offspring phenotype to the genotypes, and finally give the proportions. So if we have a look at this example, with our round and yellow, round is dominant and yellow is dominant, wrinkled and green are recessive. And in this example, although we've got the 2 dominant traits, they are homozygous dominant. And they would have to tell you that in the question, or they would have to give you information so you could work it out. So then we'd have a look at our gametes, and each gamete should have 1 copy of each gene. So you need 1 of the letter R's for the shape, 1 of the letter Y's for the colour. Now in this instance, that would mean that all of the offspring would be heterozygous, and you'll always see this pattern. When a homozygous dominant parent is reproduced with a homozygous recessive parent, you will always get 100% of the offspring being heterozygous and therefore showing the dominant trait for both of those genes. Now if we cross the F1 generation though, where both parents are heterozygous, there are now 4 possible different gametes for both parents. And this would be where we get these large Punnett squares where we put every possible gamete for both parents in the outside box. Then we work out the offspring genotypes, and then we work out all of those phenotypes, and I tend to put it in the box, so you are then definitely getting that mark of matching the phenotype to the genotype. So our ratio here is 9:3:3:1. And if you have a dihybrid cross where both parents are heterozygous for both genes, then it will always be 9:3:3:1, unless there is autosomal linkage or there is crossing over. So let's have a look then at crossing over and how this could affect the ratio. So crossing over results in new combinations of alleles in the gametes. So that means the predicted gametes that we had in our Punnett square may actually differ. So autosomal linkage is when 2 genes are located on the same chromosome, but it won't be the X or Y chromosome. So we can see here we have gene 1 is found on the same chromosome as gene 2. And this is showing us the homologous pair of that particular chromosome. So what this means is that the alleles for each gene are linked on the same chromosome. So they have to be inherited together, because in meiosis, when gametes are made, that whole chromosome will be pulled to create one particular gamete, and this chromosome will then be pulled to the other pole to create the other gametes. So that would mean you would have to have the 2 dominant alleles for the Y and R gene inherited together, and the 2 recessive alleles for the Y and R gene inherited together, as those would be the only 2 types of gametes that could be made. So if we go back to the dihybrid example where we said we have 2 parents that are both heterozygous, we said these were all the possible gametes that could be made. But if there was autosomal linkage, even though we have 2 heterozygous parents, because the R and Y gene are linked on the same chromosome, and we can see we have the 2 dominants and the 2 recessive alleles on individual chromosomes, that actually means the only possible gamete would be one that has the dominant R and Y and one that has the recessive R and Y. So our possible genotypes of the offspring and the phenotypes are here, and we get a very different ratio, a ratio of 3:1, and that then does not fit the typical 9:3 to 3:1 ratio. So that is how autosomal linkage could prevent you from getting that ratio. But we also need to consider crossing over and autosomal linkage impact as well. So we've just talked through this example where, because of the autosomal linkage, we have a 3:1 ratio, but you could actually have this scenario here. scientists actually observed the following results. So we have 4 different phenotypes, not just 2. And we can see we mainly had yellow and round, and the least common was the green and round. And this is only possible because crossing over has occurred to create new combinations of gametes. So not just these 2 gametes that we said would be possible, new combinations have been made, so there's now actually more gametes. So this is how it would have happened. We can see that we said we'd have the 2 recessive alleles linked and the 2 dominant alleles linked. If we then have crossing over, we can see that the recessive R and the dominant R cross over, that bit of the chromatid will break and exchange, and we now have 4 possible gametes instead. Now you also need to be able to see how chi-squared, the statistic, can link to inheritance. So chi-squared is used when you're investigating differences, but it's when your data is based on categories and frequencies. So if we go back to the very first example we had in this video, this is what we were looking at, the probability that 2 carriers would have a girl with cystic fibrosis. And we said we would expect the probability to be 12.5%. Now, chi-squared is the statistic used to investigate the differences between frequencies, but more specifically to determine whether there is a significant difference between the frequency we expect from our Punnett square and the frequency you actually observe. So let's go through an example. Corn can be purple or yellow and smooth or wrinkled. It's not actually just yellow like you see in the supermarkets, and this is determined by genetics. So using a Punnett square and by counting the actual number of each type of kernel, we can then use chi-squared to see, is there a difference between the expected frequency and the observed frequency? So does the frequency of the purple and yellow corn match the Mendelian genetics you would expect, which basically means the expected value you get from a Punnett square. So purple is dominant, yellow is recessive. And we've got this particular cross from 2 parents, and we would expect from this that we would get a 3-to-1 ratio of purple to yellow. A student then counted the actual number of each coloured kernel, and they recorded 21 purple and 13 yellow. So we can then use the chi-squared to see, do we have a difference between the expected ratio and the observed ratio? So we always state that there is no significant difference between the expected and the observed frequencies in the null hypothesis. And what we'd first have to do is convert the ratio, which is what we use in the Punnett square, into an actual expected frequency. So this is what was observed. So the total number of corn was 34. So if we have a 3-to-1 ratio is what we expect, the frequency that we would expect from that value would be 25.5 and 8.5. We would then do our observed minus expected. We square that, divide that by the expected, and we get our chi-squared value. In this example, the number of degrees of freedom is 1 because it's the number of categories minus 1. And we have 2 categories, purple or yellow. So the last thing you would need to do would be compare your chi-squared value to a critical value. And in this case, the calculated value of chi-squared we said was 3.176. And our critical value, we have 1 degree of freedom, so we need to look across at the row 1 and we go to 0.05, and that would be telling us that there'd be 5% probability, the results are due to chance. We always compare at this point to be able to say if we have a significant difference or not. And in this case, we can see that our calculated is less than the critical value. So that means there's more than 5% probability the results or the difference between the observed and the expected is due to chance. So that means we have to accept the null hypothesis, which was that there will be no difference between the expected and the observed frequency. And what that means is the expected frequency that we calculated using our Punnett square was the same as the frequency we observed. Or in other words, it did follow the Mendelian genetics that we expected. Now, multicellular organisms have a range of specialised cells, and those all originated from stem cells. And stem cells are undifferentiated cells that have the ability to continually divide and also become specialised. There are different types of stem cells as well, and this is determined by the differentiation abilities that they have. So you can have totipotent pluripotent, multipotent and unipotent cells. So if we have a look at the totipotent first, these stem cells can divide and produce any type of body cell. So they have the ability to differentiate into any cell. And the totipotent cells occur only for a very, very limited time in the early mammalian embryos. Pluripotent stem cells are also found in embryos, and they can divide into almost every cell, but not the placenta cells. So they can divide an unlimited number of times as well, and they're used in treating human disorders. The multipotent and unipotent stem cells are found in mature mammals, so adult cells, and can divide to form only a limited number of different cells. Unipotent can only become one type of cell. Multipotent is a very limited number. So the bone marrow is multipotent stem cells. It can only make blood cells, for example. And unipotent stem cells, an example is making cardiomyocytes. Now, there are quite a few ethical issues with using pluripotent stem cells from embryos, and that is why induced pluripotent stem cells are now used. So these can be produced from adult body cells, that's what somatic means, using protein transcription factors to overcome the ethical issues. So what you would do is take a sample of the adult cells, which we can see here, a sample from the liver, and if you switch the genes off that made those cells specialized, then you can turn those cells back into their pluripotent state. So this links to the theory of transcription factors. And that leads us into transcription factors. So this is how transcription is controlled. We'll be looking at how translation is controlled later. So in eukaryotes, the transcription of target genes is stimulated or inhibited when specific transcriptional factors are moving from the cytoplasm into the nucleus. And when they do that, they can bind to receptors into the DNA to either turn on or off genes. And what that means is that gene will either be transcribed or it won't, and therefore the protein will either be coded for and created or not. And it's this turning on and off genes which makes cells specialized. So how this works then? Transcription of a gene only occurs when a molecule from the cytoplasm enters the nucleus, because it has to bind to the DNA, and the DNA is only found in the nucleus. These molecules are proteins, and each one can bind to a different base sequence on DNA, and that will be how it initiates the transcription of a gene. Once bound, that is when transcription can then begin, and mRNA is created. That is then used in translation to create the polypeptide chain. So if the transcription factor does not bind, the gene is inactive, meaning transcription won't occur and the protein won't be made. So what we can see here is a transcription factor. So we have our DNA binding site. So this is the part that will bind onto the DNA. And we have a receptor here because often another molecule has to bind to the transcription factor before it can work. And that is where estrogen comes in. Estrogen is a steroid hormone that can initiate transcription. The way it does this is, first of all, because it's steroid based, which is a lipid, it can diffuse through the cell surface membrane into a cell. It will then bind to the receptor. So estrogen is complementary in shape to the binding site of the receptor on the transcription factor. When it binds, it causes the DNA binding site to slightly change. That transcription factor will then move into the nucleus, and because the DNA binding site has changed shape, it's now complementary to the DNA so it can attach and initiate transcription. mRNA is then created, which can then leave the nucleus and be used in translation. Now, another way that protein synthesis is controlled is through something called epigenetics. And epigenetics is the heritable change in a gene function but the DNA base sequence itself is not changed at all. Now, these changes can be caused by the environment, so things like your diet, stress levels, exposure to certain chemicals, and this can inhibit transcription. So one type of impact is increased methylation of DNA. Now, what this means is if you have lots of this chemical tag, methyl groups, binding to the DNA, it actually inhibits transcription. And the reason for that is when the methyl groups are added to DNA, they attach to the cytosine base and it prevents transcription factors from binding. It will attract the proteins that condense the DNA histone complex, and in this way it prevents sections of DNA from being transcribed. So that's what we can see happening here. The methyl groups are added, and because they are an opposite charge to the DNA, which is slightly negative in charge, it causes it to attract and to coil and bind up more tightly. And because that DNA is coiled up more tightly, there is no space for the transcription factors to be able to bind onto. And that is how it will inhibit transcription. Now, the opposite is acetylation, and that is when acetyl groups are added. But they don't bind to DNA, they actually bind to the histone protein, which is what the DNA wraps around. And decreased acetylation, so if you don't have very many acetyl groups bound to the histones, that will also inhibit transcription. If acetyl groups are removed from the DNA, then the histones become more positive. Now, just to emphasize, where it says here from the DNA, it does mean it's still bound to the histone groups, but they're within that DNA complex. But if you take away the acetyl groups, which are negative in charge, that means the histones themselves are more positive, and they'll then be able to attract the phosphate group on the DNA more, which is negative. And that makes the DNA and the histones more strongly attracted and associated, and again it coils up more tightly, preventing the transcription factors from being able to bind. So decreased, if you don't have very many acetyl groups bound, you also have transcription being inhibited. So in summary, if we think about just the methyl groups, if you don't have very many methyl groups bound, then that means the DNA is less tightly coiled, so it can be transcribed. And we call it euchromatin at that stage when it isn't very tightly coiled. If you had lots of methyl groups bound, so increased methylation of DNA, that results in the DNA and the histones tightly coiling up and the transcription factors can't bind, so transcription is inhibited. If you have increased acetylation of associated histone proteins, again, that would result in the less tightly coiled active euchromatin, so transcription can occur. But if you have decreased acetylation, you have heterochromatin, which is when all of the DNA and histones are tightly coiled, so the transcription factors cannot bind, and therefore transcription is inhibited. The final way that gene expression can be controlled is through RNA interference, or RNAi. And that happens at this stage here. So the DNA has been transcribed to make mRNA. The mRNA leaves the nucleus, but we're going to be looking at how translation can be controlled or interfered with using RNAi. So in eukaryotes and some prokaryotes as well, translation of the mRNA produced from target genes can be inhibited through RNAi. And this is when an mRNA molecule that's already been transcribed gets destroyed before it's translated, and therefore it won't create the polypeptide chain. And this is done using small interfering RNA, or siRNA. So this is the molecule that is going to destroy the mRNA, and that's why this is called RNA interference, because our mRNA has been interfered with. So let's have a look at how that happens. Transcription results in the production of mRNA, and mRNA is then used in translation by attaching to a ribosome, and that creates the primary structure of a protein or the polypeptide chain. However, if there is small interfering RNA, that can disrupt the mRNA that's been created and destroy it, and therefore it prevents translation from occurring. And this is what we mean by RNA interference. So small interfering RNA results in this interference. So that's just a concept map just to show you how this all links together. Now, how this actually destroys the mRNA is you have some double-stranded RNA, but there's an enzyme that can cut that and make it single-stranded, but also into small pieces. So it's small and it's going to be interfering, so siRNA. That will then bind to an enzyme, and that enzyme's called RISC, but you don't need to know that's the name. Because it's single-stranded, this complex of the siRNA and the enzyme can bind to the mRNA molecule that was made in transcription. And that then means that the enzyme is able to cut up that mRNA into small pieces, and therefore it cannot be used in translation. It won't be able to bind to the ribosome, and because it's cut up, you can't translate that entire sequence. The next thing you need to know about is cancer, which we briefly mentioned at the start. So it can result from mutations in genes that regulate mitosis. And if these genes do mutate and a non-functioning protein is created, then that means mitosis cannot be controlled. And that is why you end up with this uncontrollable cell division, and that is when a mass of cells or a tumour is created. Now, there are different types of tumours. You can have what's called a benign tumour or a malignant tumour. And you do need to know the differences in the properties of these. So a benign tumour can grow very large still, but it will be dividing at a slower rate. So it will take longer for there to be a larger tumour. Benign is non-cancerous, and one of the reasons it's classed as non-cancerous is they produce this adhesive sticky molecule, and that will then surround it so it cannot move. It's also in a capsule as well, which means that these cells that are having this uncontrolled mitosis, they're not able to break off and spread to other parts of the body. And therefore, if surgery can be conducted on the part of the body that the tumour is located, you may be able to cut out and remove that entire encapsulated tumour, and then it'd be very rare that the tumour would regrow. So the impact is localised, and it's often non-life-threatening because you can cut it out and remove it. But it does, as I said, depend on the location of the tumour. So for example, a benign brain tumour is still very dangerous because any type of brain surgery is very high risk because you have to cut through the skull, and in removing that tumour you could actually damage some of the other brain tissue. Now, in contrast, malignant tumours, these are the ones that are classed as cancerous, and they do grow much quicker. The cell nucleus becomes very large and it returns back to its unspecialised state again. They don't produce that adhesive sticky layer or the capsule, and therefore they can metastasise. And what that means is cells of the tumour can break off, spread through the blood, and therefore lodge in other tissues in the body, and then you get secondary tumours in other organs. The other thing that we can see here is because it's not encapsulated, yes, it also links the idea of metastasis, but also it can grow projections and reach the blood supply. And once that tumour has its own blood supply, it's going to be receiving lots of oxygen and glucose for respiration, and that is one of the reasons why those cancerous tumours can grow so rapidly. It can be life-threatening, and the removal of the tumour needs often supplementary treatment like radiotherapy and chemotherapy, and the reoccurrence is more likely than with a benign tumour. So the tumour development then, as we said, it could be due to a gene mutation, and if it is a gene mutation, the mutation would have had to have occurred either in the tumour suppressor gene and or the oncogene. It could also be linked to epigenetics though. So if you have abnormal methylation of a tumour suppressor gene or an oncogene, or if you have increased estrogen concentrations affecting transcription. So those are all of the possible causes of these tumour developments. Now, we've used this term tumour suppressor gene and oncogene, but let's just go through what those genes code for. So oncogenes are the mutated version of a proto-oncogene. And proto-oncogenes code for proteins which are involved in initiating DNA replication in the cell cycle. An oncogene, though, can result in the process being permanently activated to make cells continually divide even if new cells aren't needed in that part of the body currently. Tumour suppressor genes code for proteins which are involved in controlling the cell cycle, and they're involved in slowing down the cell cycle or to cause cell death. Now, if a mutation results in the tumour suppressor gene, that means that protein isn't produced and cell division isn't slowed down, so it'd continue. Mutated cells would not be identified and destroyed. Now, we said that epigenetics can also link to the cause of tumours, and that was because of abnormal methylation. So we looked at how methylation can cause genes to be turned on or off, and if a tumour suppressor gene becomes hypermethylated, that would mean there's an increased number of methyl groups bound to it, and that results in the gene being inactive and it is switched off. So if that happened to a tumour suppressor gene, that means that protein won't be produced that slows down the cell division in the cell cycle. Now, the opposite could occur in an oncogene. So as they may be hypomethylated, meaning you don't have very many methyl groups attached, that could result in the gene being permanently switched on, and therefore lots of the protein is being produced to initiate that cell division constantly. Now, the final thing we said is estrogen also has an impact in increasing the risk of cancer, and it's linked to this specific example. So estrogen is produced by the ovaries to regulate the menstrual cycle. However, after the menopause, the ovaries stop producing estrogen, and instead the fat cells in the breast tissue produce estrogen instead. And this is what's been linked to breast cancer in women post-menopause, and that is why age is one of the risk factors of developing breast cancer. So we looked at earlier how estrogen is a molecule that can bind to receptors on transcription factors. So if that is going to happen to a transcription factor involved in the cell division, so for example, it's a transcription factor that can bind to the DNA of a proto-oncogene, that would mean that that particular gene is permanently turned on, activating cell division within the breast tissue. Now, it has a knock-on effect as well, because as the tumour grows, because those tumour cells are breast tissue cells, that will result in even more estrogen being produced, and therefore even more of those proto-oncogenes could be switched on, activating more cell division. So this is an example of positive feedback. The more that we have of the tumour, the more estrogen, but that results in even more tumour cells occurring. Photosynthesis is knowing where it occurs and just a mini recap on the structure of a chloroplast, because you need to know where each stage of photosynthesis happens. So here on our chloroplast, we can see the thylakoid stacks, and the thylakoid membrane is on the outside of that. And that's where you have these folded membranes where you have all the proteins embedded for the electron carrier chain, and also ATP synthase. And that is where the light-dependent reactions occur. A stack of them is called a granum. The fluid part in the centre is called the stroma, and that's where the light-independent reactions happen. And you have lots of enzymes there for those. And then we have the inner and outer membranes as well, which are controlling what can enter and exit. Photosynthesis is a two-stage reaction. We have the light-dependent and the light-independent reactions. And the light-dependent reactions happen first, and as the name suggests, it requires light. This happens on the thylakoid membrane, or you could call that the granum. In this light-dependent reaction stage, light energy is used and water to create ATP and reduced NADP. And those two products are required for the light independent stage, which comes second. So there's 4 key steps here. We have photolysis, or sometimes it's pronounced photolysis to remember the stage, photoionization of chlorophyll, and chemiosmosis. Now those are actually the 3 processes. Production of ATP and reduced NADP is the outcome, but AQA states these are the 4 key stages that you need to know. So starting with photolysis or photolysis, photo refers to light, lysis means splitting. So photolysis of water means light energy is absorbed and then that splits the water. And it splits it into oxygen, electrons and protons, which are hydrogen ions. What then happens is the protons are picked up by NADP to form reduced NADP or NADPH, and that is then used in the light independent reaction. The electrons are passed along a chain of electron carriers, and the oxygen is either used in respiration, but it's not used in photosynthesis. So if it isn't needed for respiration, it will diffuse out through the stomata. We'll go through in a bit more detail what happens to the protons and electrons when we get to chemiosmosis. Photoionization of chlorophyll, again, is to do with light, because we've got photo. So light energy is absorbed by the chlorophyll, and the ionization part is referring to the fact that the light energy results in the electrons gaining energy. And we describe that as them becoming excited and raising up an energy level. And that causes the electrons to leave the chlorophyll, and therefore the chlorophyll has been ionized. Some of the energy from the released electrons is used to make ATP and reduced NADP in chemiosmosis. So let's have a look at chemiosmosis then, because this then links it all together for the light-dependent reactions. So we've said that the electrons gain energy from the chlorophyll, will be released from the chlorophyll. What happens is those electrons move along a series of proteins embedded within the thylakoid membrane. And as those electrons move along the proteins, they release energy. And some of that energy is used to actively transport or pump those protons from the stroma across a protein and into the thylakoid lumen. This then results in a concentration gradient being built up, but we actually call it an electrochemical gradient because we have a charged molecule, so we have ions there. So now we have this electrochemical gradient that enables the protons to move by facilitated diffusion back down their concentration gradient to the stroma. But the only protein that they can attach to is ATP synthase. And as the protons attach and move through ATP synthase, that enables the enzyme, which the protons move through, to phosphorylate the ADP into ATP. So that's how the ATP is produced. We then have the protons back on the stroma side, and this is how we create NADP that is reduced, or reduced NADP. So the NADP coenzyme picks up the electrons from the end of the electron transport chain. It picks up the protons after they've passed through ATP synthase, and that reduces NADP, which makes it NADPH. So now we've created ATP and reduced NADP. Those two molecules are used in the light independent reactions, also known as the Calvin cycle. This occurs in the stroma. It requires enzymes, and the enzyme Ribisco is the enzyme that you need to know. And because of that, this stage is temperature sensitive. But it doesn't require light energy for the Calvin cycle to occur. Now, the Calvin cycle uses carbon dioxide as well as the two products from the light-dependent reaction, and we create a hexose sugar. The ATP that is needed is hydrolyzed to provide energy for the reaction, and the reduced NADP is used to donate the hydrogen to reduce the molecule GP within the cycle. So let's go through this cycle then. The first part of the cycle we'll look at is how the carbon dioxide enters the cycle. So carbon dioxide reacts with RUBP, and that is a 5-carbon compound. So all of these yellow circles are representing carbon molecules. So we can see we've got a 5-carbon compound reacting with carbon dioxide, which only has 1 carbon. That would result in a 6-carbon compound, but actually it's unstable, so it's split into 2 3-carbon compounds. And that's our GP. So we have 2 lots of GP. This is then where one of the ATP molecules is used, and it's where the reduced NADP is used. GP picks up the hydrogen, and therefore NADP is reformed, and the GP is now reduced to form TP. That reduction requires energy from ATP. The TP is then used for 2 things. One of the carbons is removed from these 2 times 3 carbon compounds, and it goes towards creating a hexose sugar. That will leave behind 5 carbons, but as a 3-carbon compound and a 2-carbon compound. So ATP is needed to form those back together, or in other words, regenerate RUBP. Now, that is just one round of the cycle, and that only created 1 carbon, which isn't enough for a hexose sugar. Hexose means 6. So the Calvin cycle has to happen 6 times before a hexose sugar is made. And that could be glucose, it could be sucrose, and then the plant can use that glucose or sucrose and convert it into whatever organic compound is needed. So that could be another carbohydrate, so cellulose for the cell walls. It could be a store of glucose in the form of starch, or it could convert the hexose sugar into glycerol and then combine with fatty acids to make lipids, or it could combine it with nitrates to form the amino acids. Now you need to know a little bit about limiting factors as well, and the limiting factor is anything that reduces the rate of photosynthesis. And that could be light intensity, carbon dioxide concentration, or temperature. And the way you can look at a graph to tell what is currently limiting the rate of reaction is wherever you have a positive correlation on the graph, that tells you that whatever is on the x-axis is currently limiting the rate. So that means up until this point, or probably a bit further up to here, light intensity is limiting the rate of reaction. And we know that because as we increase the light intensity, the rate increased. But that then starts to plateau and level off. So even though you add more light, the rate doesn't increase. And what that means is there must be another factor that is now limiting the rate. And that must be either carbon dioxide concentration or temperature. So it's the same concept with carbon dioxide concentration, that as the rate is increasing and the carbon dioxide concentration is increasing, we know that carbon dioxide is the limiting factor until it plateaus. Temperature, this is linked to the idea of enzymes because it's an enzyme-controlled reaction. So at low temperatures, the rate of reaction is lower because we don't have enough successful collisions, there's not enough kinetic energy, we don't get enough enzyme-substrate complexes. But if it gets too hot, the enzyme denatures. Now the carbon dioxide, if you had to say why that limits, that is because it is a reactant in the Calvin cycle. Light intensity can limit the rate of reaction because light energy is needed in the light-dependent reactions for photolysis and photoionization. So for maximum photosynthesis, in agriculture, when they're growing crops, they need to consider how they can remove those limiting factors to maximise their profits. So sometimes you get application questions linked to this. So the idea of adding in artificial lighting, heating a greenhouse, and that could be through burning fuels because that will also produce carbon dioxide. But all of those additions cost. So it has to be considered whether the cost of the additional light, heat or fuel is offset by the additional profit. So they have to make sure it's cost effective in order for it to be worthwhile to pay for those additional conditions.